Here in this third and final unit part of Unit 4, we will discuss various conditions and disorders related to carbohydrate metabolism, and the most prevalent one here is diabetes mellitus. And diabetes mellitus is a disorder of carbohydrate metabolism that's defined by two key signs here. That's hyperglycemia, a higher than normal amount of glucose in the blood, and glycosuria, a higher than normal amount of glucose in urine. And this results from the inadequate production or utilization of insulin. And we'll discuss these in a little bit more detail as it relates to the different types of diabetes mellitus. Over 2 million Canadians are estimated to be afflicted with diabetes mellitus. 1 in 10 of those afflicted have what's referred to as type 1 diabetes mellitus. An older term that used to be used as insulin-dependent diabetes mellitus, while 9 in 10 are afflicted with what's called type 2 diabetes mellitus, or what used to be referred to as non-insulin-dependent diabetes mellitus. A third type of diabetes mellitus that we'll discuss briefly at the end of this unit is gestational diabetes, which is the same disorder of carbohydrate metabolism that results as is created as a result of pregnancy. Approximately 4% of all pregnancies result in gestational diabetes. And that prevalence is higher among Aboriginal women. But coming back to the basic definition of diabetes mellitus, diabetes is a Greek term, a Greek derivative meaning a passing through, and this is a term usually characterized by excessive urination. The term mellitus is a Latin derivative meaning sweetened with honey, and that indicates the presence of sugar, or rather glucose, in the urine, and that's one of the key diagnostic features of diabetes mellitus, is a greater than normal amount of glucose in the urine. A separate disorder that we will not be discussing in this unit but worth mentioning is diabetes insipidus, and this is a condition that is involved with the production of excessive urination, hence the term diabetes, but the excessive urination is related to impaired water reabsorption in the kidney as a result of a lack of antidiuretic hormone, also referred to as vasopressin, which is a hormone that, when secreted, produces an increase in water reabsorption in the kidney back into the cardiovascular system. But we'll start by examining type 1 diabetes mellitus, and this is a condition that results usually as an autoimmune disorder. In other words, the beta cells in the pancreas that produces insulin are ingested by the immune system. So usually early in life, as a result of some type of viral infection, a person's immune system will not only try to attack that viral infection, but will also attack healthy cells. And the result of this, if these beta cells are destroyed, without the beta cells, the person does not have the ability to produce the protein insulin, which functions as a hormone, and that leads to hyperglycemia. Recall, insulin is the hormone that triggers the removal of glucose from the cardiovascular system and promotes movement into liver cells and skeletal muscle cells. So the normal range of glucose in the blood, depending upon the scale, is between 80 and 110 milligrams per deciliter of blood, and after a carbohydrate meal it may rise to 140. So 140 is a normal value after a carbohydrate meal. However, if a person has not eaten for 6 to 8 hours, 140 milligrams of glucose per deciliter of blood is an extremely high value, and would indicate this person is afflicted with diabetes mellitus. So without insulin, we don't have the signal for glucose movement into liver cells and skeletal muscle cells. So continuing from the previous slide, so without insulin, we don't have the glucose uptake, and so the consequences of this is that we don't have any translocation of GLUT4. So recall in part 2 of Unit 4, insulin caused the protein GLUT4 to move into the membrane to assist with the removal of extra glucose molecules from the cardiovascular system. Insulin is also an activator of GLUT2, same protein, same structure in the liver, but without insulin, GLUT2 is not activated, and if GLUT2 is not activated, glucose does not move into the liver. So without glucose being brought in, the liver senses that the person is in a low blood glucose state. So the liver actually makes a mistake here without insulin and interprets the lack of insulin as low glucose in the blood, where it actually is higher than normal. So that would increase the rate of what's called glycogenolysis. This is the breakdown of liver glycogen, so that the individual glucose molecules are separated from each other in its storage form and is pushed into the cardiovascular system and actually makes an existing hyperglycemic state worse. And with the increase in glucose concentration in the blood, that blood will eventually get filtered as a normal process by the kidney, and the renal threshold reflects the maximum amount of glucose that can be reabsorbed back into the cardiovascular system after filtration by the kidney. And so after the kidney hits that renal threshold, those extra glucose molecules are not reabsorbed into the cardiovascular system, and they're excreted from the body in the urine, hence the increase in glucose concentration in the urine. And so this results, as I mentioned, as exceeding the capacity of renal tubules, the functional units in the kidney, to reabsorb those extra glucose molecules. With the extra glucose molecules that are not reabsorbed, those glucose molecules will attract water molecules, and that will result in the excessive production of even more urine. So a telltale symptom of type 1 diabetes is consistent thirst and frequent urination. And these were signs and symptoms that were first observed over 2,000 years ago. The risk factors for developing type 1 diabetes mellitus, there's a genetic susceptibility, so people that test for a particular gene that is mutated has a increased risk of developing type 1 diabetes mellitus, and a family history of type 1 or type 2 diabetes mellitus does increase the risk of developing type 1. So in terms of energy utilization, which becomes quite problematic in people afflicted with type 1 diabetes mellitus, recall glucose cannot get into the cells, muscle cells as well as liver cells. So the body senses that there's a starvation state. So this sensation by the body is actually a mistake. This is a misinterpretation of the fact that there isn't a lot of glucose available to these cells. So the cells in the body will have no choice but to find another fuel source. So fatty acids are actually going to be the fuel of choice initially. So without insulin, We don't have 7-TM receptor binding. 7-TM is a protein receptor on fat cells that will trigger the activation of triglyceride lipase. So insulin has the effect of inhibiting triglyceride lipase, hence insulin having a fat storage effect. But without insulin, there's no inhibition of triglyceride lipase. So all the stored fatty acids, I shouldn't say all the stored, but many of the stored fatty acids in fat cells are broken down and released into the cardiovascular system. And so this results in an increased utilization of fatty acids as a fuel. But because fatty acids are too large to move from the cardiovascular system and over to the nervous system, the body will eventually start to consume muscle protein and break down the protein to make new glucose molecules. So the body attempts to conserve muscle protein, but eventually in a person with type 1 diabetes mellitus that becomes advanced, this will lead to excessive muscle wasting. So the complications that are related to insulin deficiency, because the beta cells have been destroyed in the type 1 case, is there's an increase in the adrenaline hormones, also referred to as catecholamines. And so if there's a chronic increase in catecholamine production and secretion, that has the effect of increasing overall metabolism in the body, increasing the metabolic rate. There's an increase in cortisol secretion, and cortisol is a stress hormone that carries out many functions. One function is that it accelerates muscle protein breakdown, and with the increase in glucagon secretion, glucagon will have the effect of making the hyperglycemia worse by causing the liver to release more glucose into the cardiovascular system. Insulin deficiency, without insulin, the inhibitory effect on the enzyme that breaks down fatty acids in fat cells is not there, so that will increase lipolysis, the breakdown of stored lipids. And because lipid breakdown increases, muscle protein breakdown increases, a byproduct of those is an increase in these small 2-carbon compounds called keto acids. These are very acidic compounds that have the effect of decreasing body pH that will lead to number 4, a reflex increase in breathing rate, referred to as Kussmaul respirations. And that results in a form of hyperventilation. So the overall complications that relate to that are related with type 1 diabetes mellitus and hyperglycemia. So with a chronically high level of extra glucose molecules in the cardiovascular system, one of the effects is an increase in blood pressure. And that's due to the fact that there are more solutes, more glucose molecules, and more H2O molecules bound to those glucose in the blood. Because the blood vessels coursing through the kidney are very small, it can lead to kidney disease. Because of the increased pressure, there's increased filtration, and renal tubules become more active, placing more stress on the kidneys in a chronic fashion. The increased blood pressure can lead to various peripheral vascular disease complications, and because of this high blood pressure, the very small blood vessels, the capillaries that feed blood to the feet, the neurons that are and the sensory neurons in the feet and the eyes can rupture and will lead to peripheral neuropathies and blindness in advanced stages. An acute complication that people afflicted with type 1 diabetes mellitus should be concerned about is something called diabetic ketoacidosis. And so this is an excessive production of keto acids that will decrease the pH in the body, and it's a severe form of hyperglycemia and can be a life-threatening situation. So some of the symptoms in a person that is experiencing ketoacidosis is nausea, vomiting, stomach pain, a fruity smelling breath, and anywhere from 2 to 5% of these cases are potentially fatal. And people with type 1 diabetes mellitus, because of the inability to produce their own insulin, are required to take an external dosage. And so there are a number of different products on the market for obtaining an exogenous insulin supply. And so Humulin is made by the Eli Lilly company, and there are various types of insulin products. There are short-term release type of insulin injections, or long-term, slow release, depending upon a person's diet, depending upon their physical activity. Biosynthetic insulin is actually manufactured from genetically modified E. coli bacteria, and so that provides manufacturers with sufficient amount of insulin to package and to deliver to those that require it. Even though a person is taking insulin, these individuals do require blood glucose monitoring throughout the day, and the type of insulin, the frequency of injections will depend upon a person's eating pattern, the level of exercise or physical activity, quantity and quality, and really the timing of all three factors. Another type of insulin, Humalog, is a mealtime insulin. So it's a very fast-acting type of insulin that a person would inject at the time of a meal. Now, the most common type of diabetes mellitus, 9 out of 10 Canadians so afflicted, is what's referred to as type 2. Older term used to be non-insulin dependent diabetes mellitus, but that term is no longer used. It also used to be referred to as adult onset diabetes mellitus, but in the last 20 years, we're seeing younger and younger people, children, developing this type of diabetes mellitus. In fact, studies suggest that 1 out of every 3 children in the U.S. and Canada born in 2000 will likely be diagnosed with type 2 diabetes in their lifetime, which makes it quite problematic. First Nations children are particularly at risk, and children as early as 8 years old have been diagnosed with type 2 diabetes mellitus, and generally this is related to the increasing rate of obesity in children in the last 2 decades. So in terms of type 2 diabetes mellitus, it carries the same diagnosis as type 1. It's characterized by a chronically high level of blood glucose under fasting conditions. So recall, after a meal, it is normal that blood glucose increases, but we expect those blood glucose levels to go down after a fasting period of 6 to 8 hours. But we see here in a person that is afflicted with type 2 diabetes mellitus, although the signs and symptoms here, the diagnostic criteria are the same, this type of diabetes mellitus is not characterized by destruction of beta cells. In fact, a person afflicted with type 2 has an intact insulin supply. But the problem in this type of condition is that the glucose using cells, the muscle cells, liver cells, are what are referred to as insulin resistant. So although a person is able to make their own insulin, the muscle cell is not responding to that insulin and activating the proteins to help pull the glucose molecules out of the cardiovascular system. So normally, Insulin will cause GLUT4, going back to our part 2 of this unit, to move into the muscle membrane surface. This is the normal function of insulin. But in a person afflicted with type 2 diabetes mellitus, this does not happen, not as readily. Muscle contraction will also have a similar effect as insulin in translating GLUT4 to the cell membrane, making physical activity or exercise an effective way for a person afflicted with type 2 to manage their condition. So the management here, the first strategy here is related to eating pattern. So it's recommended that a person reduce simple carbohydrate intake, so reduce sugars, the intake of sugars, focus more on complex carbohydrates, and physical activity. So increasing a chronic level of physical activity will have the effect of making the muscle cell more sensitive to moving GLUT4 into the membrane, and that's going to improve tissue glucose uptake from the cardiovascular system. There are some statistical risk factors that puts a person at greater risk for developing type 2 diabetes mellitus. So a family history of type 2 suggests that there's some genetic predisposition to developing type 2. Statistically, people that fit the overweight or obese category are at greater risk for developing this problem related to glucose handling by the body. A sedentary lifestyle, low HDLs or high triglycerides. So in the blood, these are also considered to be risk factors for developing type 2 diabetes mellitus. A woman that develops diabetes mellitus as a result of pregnancy is at a statistically greater risk for developing type 2 diabetes mellitus later in life. These are statistical risk factors. They're not necessarily cause and effect, but factors that people should be aware of. Syndrome X, also referred to as metabolic syndrome, is a syndrome that results from a combination of a greater than the normal amount of insulin in blood, hyperinsulinemia, hypertension, blood pressure, an increase in blood pressure, and insulin resistance. So when we think about type 2 diabetes mellitus, it usually comes along with other conditions that relate to more than the normal amount of insulin and high blood pressure or even cardiovascular disease. A significant correlation of developing these conditions is body weight. And so again, these are statistical correlations that people that fit the overweight or obese category are more likely to develop this metabolic syndrome, and a person that presents with these conditions are most likely in what is referred to being in a pre-diabetic stage. In terms of gestational diabetes, as mentioned earlier, this is a development of the same condition discussed, hyperglycemia, glycosuria, as a result of pregnancy. Approximately 4% of all pregnancies will develop this condition after the first trimester. And this results later on with a higher incidence of becoming pre-diabetic later in life, but again, that's a statistical relationship, or a statistical relationship, not cause and effect. There are a number of different theories as to why a woman would develop gestational diabetes in pregnancy. One theory is that the increase in cortisol secretion, it's the stress hormone of the body, and that has the effect of increasing insulin resistance by various cells. The increase in secretion of progesterone is thought to be inhibitory of beta cells in the pancreas. A hormone produced by the placenta, human placental lactogen, is also hypothesized in the insulin resistance nature of this type of diabetes mellitus. So in the case of gestational diabetes, a woman is secreting the normal amount of insulin. It's just that the muscle cells and liver cells are not as responsive. So this is characteristic of type 2. So the risk factors include a woman that has an existing type 2 diabetes mellitus, or type 1 for that matter, family history, a woman whose body mass index would put them in the overweight or obese category. Women that become pregnant at an older age are statistically at risk for developing a gestational diabetes, and women within various ethnic groups have all been observed to statistically be at greater risk for developing gestational diabetes mellitus. So the complications are similar to type 2 diabetes mellitus and type 1. The first complication, the most pressing one, is high blood pressure. Recall that in a hyperglycemic state, there is an increase in the number of solutes in plasma, and that has the effect of putting pressure from inside the cardiovascular system. But because of the pregnancy, another complication is macrosomia, in which which results in a large birth weight for the newborn. Recall that newborns, the developing fetus, feeds on glucose, and because there's an excessive amount of glucose in the blood, that will have the effect of accelerating the growth of the cells in the growing fetus. And that does pose an increase in certain health risks during the delivery. In severe cases, it can result in jaundice for the newborn, which may result from an excessive breakdown of red blood cells. It can affect the brain and growth development in the newborn, which is why this type of condition, women that do develop gestational diabetes mellitus, are followed quite closely and are monitored on a daily basis. Now, the brain and growth development in newborn can result from hypoglycemia. So why would hypoglycemia result? In response to the high levels of glucose in the blood, the body may produce excessive insulin that could, for a short period of time, have the effect of decreasing blood glucose for a short period of time to a level that would not be sufficient for all the developing neurons to survive. So this is managed with the typical approach with diabetes mellitus from a dietary perspective, focusing on complex carbohydrates, frequent eating to prevent large swings in blood glucose levels, reasonable levels of physical activity so that the activation of the muscle cells will help promote the movement of the glucose transport proteins into the membrane. Daily monitoring of blood glucose levels multiple times per day is absolutely necessary in this case. And then post-birth monitoring is usually followed up because statistically a woman that develops gestational diabetes mellitus is at greater risk for developing type 2 later in life. It's not cause and effect. It's a statistical relationship that women who develop gestational should keep an eye on. So this ends the third part of our Unit 4 on carbohydrates.